Hello everyone, we are going to discuss a new topic, that is the restriction enzyme. Okay, so this is the content of this presentation, that is the introduction, discovery, how the restriction enzyme works, types of restriction enzymes, the subunits of restriction enzyme, nomenclature of restriction enzymes. Okay, recognition, sequence of restriction enzyme, properties of restriction enzymes, and applications of restriction enzymes. Okay, what we mean by the restriction enzyme? Restriction endonuclease are the enzymes which are responsible for the production of internal cuts called cleavage in the DNA molecule. Okay, called by the restriction endonuclease. Restriction endonuclease is a bacterial enzyme that cuts the double stranded DNA into the fragments of logarithmic recognize specific nucleotide sequence known as recognition or the restriction sites. Okay, so restriction enzyme is an protein which is responsible for the cutting of the DNA at its specific points. Okay, restriction enzymes are believed to be involved by the bacteria to resist the heredity. So bacterial cell have its own immune system, okay, or attacking system on the foreign particles. Okay, meaning that these restriction enzymes play an important role, okay, to resist its attack on the bacterial cell. So restriction enzymes are also known as the molecular scissors. Okay, discovery of restriction enzymes. That restriction enzyme originated from the studies of phage lambda. Okay, phage lambda is a bacteriophage. Bacteriophage means virus which infects the bacteria. In 1960 was shown by the Werner Erber, okay, and Matthew Messelsen's, but they studied only one type of restriction enzyme. Okay, so in 1970 Hamilton O. Smith, Thomas Kelly, Cantwell Cox isolated the characterized the first type of restriction enzymes in second from the bacterium Haemophilus influenza. Okay, this is the next restriction enzyme which is discovered by the O. Smith and Thomas Kelly. Okay, for their work in the discovery and characterization of restriction enzymes in 1978, they get Nobel Prize for the physiology or medicine was awarded Werner Erber and Matthews and Hamilton O. Smith. Means this is one of the most milestone research in the field of life science. Okay, so restriction enzymes is leads to the formation of a new era, formation of a new milestone in the field of molecular biology. Okay, the types of restriction enzyme, there are three basic types of the restriction enzyme: type 1, type 2, and type 3 restriction endonuclease. Categorization of restriction enzyme on the basis of their composition, coenzyme requirement, the nature of target sequence, the position of DNA sequence, DNA cleavage site related to the target sequence. Okay, how the restriction enzyme work? The restriction enzyme can recognize a specific sequence of nucleotide and produce a double strand cut in the DNA. These cut are called of two types. So restriction enzyme have its own mechanism of work. They produce two various types of end, that is the one is the blunt end and another one is the sticky end. Blunt end is a direct exact cutting of the both strand of DNA at the same nucleotide, and sticky end is produced by the in the zigzag manner. Okay, blunt end means these blunt ended fragments can be joined to any other DNA fragments with the blunt ends. Okay, certain enzymes are used to certain types of DNA cloning experiment. Means blunt end also important in the some type of cloning experiment. But the most useful ends are the sticky ends. Okay, DNA fragments with the complementary sticky ends can be combined to create new molecules, which allows creation and manipulation of DNA sequence from the different sources. If we want to join the two or more DNA sequence of various different species, we can use this enzymes which are responsible for the production of sticky ends. Okay, so one of the most important enzymes used in the recombinant DNA technology are the enzymes which produce the sticky ends of the DNA. Okay, this is the diagram which shows how the eco R1 cuts the DNA. Okay, so one of first type is the type first restriction endonuclease. Type first restriction endonuclease are complex endonuclease have a recognition sequence of 15 base pairs. This is recognized at a 15 base pairs sequence and they cleave around 1000 base pair away from the 5' end of the TCA located within the recognition site. Means recognition site. From the recognition site, it cleaves away 1000 base pairs from the its TCA site. So, example is EcoK12 EcoB. Okay. Types first restriction enzyme work with the help of many cofactors like adenosine, methionine, hydrolyzed adenosine triphosphate, that is the ATP, and magnesium. Okay. The type restriction enzyme methylates the DNA sequence at the site of recognition and type first. Okay. Then subunits of type first, that is SSDR, which is required for the restriction, HSDM, which is necessary for the addition of liquid group to the host DNA, that is the methyl transferase activity, and HSDS, which is responsible for the methyl transferase activity. Okay. The second type of restriction endonuclease, which is a mostly common used enzyme, which is in the field of molecular biology, in the field of recombinant DNA technology. Okay, remarkably stable and induced cleavage either in the most cases within the or outside their recognition sequence, which is the asymmetrical. The type of DNA restriction enzymes are work with the help of only single cofactor like a SAM. Okay, the type second also various features. The type first type of enzyme to be isolated and second in 1970. Okay, which the type second restriction enzyme can generate two different types of cuts depending upon whether cut both stand at the center of recognition sequence. Okay. The subgroup of type second, type B, EMT. Okay, type third recognition endonucleases. Type third restriction enzymes are intermediate between the type first and type second endonuclease. They cleave DNA in the intermediate vicinity of their recognition sites. Eco P1, Eco P15, its example. They cut DNA up to 30 to 20-30 base pair away from the recognition site. Okay, their cutting is in between the 20 to 30 base pairs. Okay, this enzyme contains more than one subunit. Okay. Then the nomenclature of restriction enzymes, it depends upon its genus, species, strain, and isolated order. So, for example, Eco R1, the genus is Eco Escherichia, species is called strain R, and order of discovery is the first. Okay. Properties of restriction enzymes, nature of enzyme. It shows, that is the first type. It shows endonuclease and methylase activity. Second, it separate endonuclease and methylase activity, and third, it shows the endonuclease and methylase activity. Protein structure, three different structure of subunit. In the type of first, type second, two identical subunit is there, and type third, two different subunit is there. Okay, restriction requirement, ATP, MD2, and SAM, that is adenosine, methionine, and type third is the required. Type second is required MD plus 2, which is the most prominent used in the recombinant DNA technology or molecular biology. Type third is the ATP and MD2 plus. Okay, so random up to 1000 base pair near to the target site and 24 to 26 base pair to the restriction site. Example of type 1 is Eco B, type 2 is Eco R1, and type 3 Eco B1. Okay, application of restriction enzyme, they are used in the gene cloning experiment, floating expression experiment. So recombinant DNA technology has a wide use of the restriction enzymes, which is used in the creation of novel features by using the DNA. Restriction enzyme are used in the biotechnology to cut the DNA into smaller strands in order to study the fragments with length different among the individual, that is the restriction fragment length polymorphism, which involves the separation of DNA fragment on the basis of their molecular weight. Okay, is this of this method different from the use of agarose electrophoresis for the separation of DNA fragments? It provides another application provides the different way of manipulating DNA, such as the creation of recombinant DNA, which has endless applications. Okay, allow for the large scale production human insulin for diabetic insulin, E. coli, as well as the hepatitis B or HPV vaccine. Okay, cloning of molecules and studying the nucleotide sequences. These are the various applications of restriction enzymes. Okay, I think all of you get it, my presentation. Okay, any query you can call me or message me on my number. Thank you.